Let's go ahead and get started. First things first, there will not be clicker questions today, so if those of you need to leave, you can go and do that now. Second thing is that the clicker questions that were done before, as long as they got recorded, I will be including those, and there will be clicker questions on Friday based on today's lecture. Which brings me to what you would like me to do on Friday. There are really two options. One is to do what I usually do, which is basically review all of the lectures since the last midterm. So those would be mine and our guest lectures that we had last week and on Monday. Or would you prefer me to concentrate just on the ones that we had last week and Monday? So I see some nodding, and so I will actually, I probably should have had a clicker question for this so I could actually get your votes. But we'll do old-fashioned hands instead. How many people would like me to cover everything on Friday? Okay, how about just the guest lectures on Friday? Okay, there's a lot more of those. Okay, so I will do really concentrate on the guest lectures on Friday. I've also put together today's lecture, I should say, based on what the guest lecturers were able to go over and some of the things that I thought were particularly important sort of towards the end of their lectures. And you'll see that in the format of the slides today. So I want to start out talking a little bit about the so-called two-hybrid screen. This is, in fact, a really good review about transcription factors, which are usually transcriptional activators, and how they actually function. And it's a really nice tool that you can use to really screen through multiple different proteins that you have and what you think are potentially interacting with each other. And hopefully that's something which has come up over the last, what has it been, almost 10 weeks, that really protein-protein and protein-DNA interactions are really critically important for how molecular biology works, how these molecules actually interact with each other. We'll talk a little bit about nuclear receptors. It turns out that nuclear receptors are a whole class of transcriptional regulators. They can function both in a repressive form or an activating form and are really, really important for development, particularly in metazoans like us. Shift gears a little bit and then talk a little bit about imprinting. This has to do with DNA methylation. And unfortunately, it's a topic which is not, I think, terribly well covered in the textbook. But I did want to talk about it a little bit. It's one of the other parts of epigenetic inheritance that I wanted to talk about. X-inactivation is another nice example of epigenetic inheritance. But here, it's really based on RNA. And so one of the take-home messages from this course should hopefully be that RNA is probably at least as important as proteins, certainly at a regulatory level, and it's been ignored for many, many years. People always used to think of mRNA, it's the messenger. It's what you take going from your DNA going to protein. It's just this thing that happens to be in the middle. There's a lot more going on than just messenger RNA. Talk a little bit about the processing, which I think is something, again, that our guest lecturers didn't get to, and messenger RNA export. So I'll start out with the two-hybrid screen. Basically, the whole principle of the two-hybrid screen takes advantage of the fact that transcriptional regulators are modular. And when I say modular, that means that they have at least two, and often sometimes many more than that, different domains. And so here's an example of a transcriptional activator. If I can get my mouse to work here. With a DNA binding domain, this is the purple thing down here at the bottom. Very often have linker regions between the two, usually very flexible, and then a separate activation domain. And these two can really function independently of each other. And so there's a lot of mixing and matching that can go on. And we talked about this when we talked about genetic recombination, where you can mix and match various different domains, usually through homologous recombination. One particular example of that is the Gal4 protein. This is a transcriptional activator from Saccharomyces cerevisiae. It has a number of different domains. Here is a zinc hormone-binding domain, which serves as its DNA binding domain. You have a dimerization domain. Many of these are dimers, and hopefully Dr. Bartlett talked a little bit about dimerization. We'll talk more about that today. And then, really importantly, is this activating region. In fact, there are two different activating regions and one specific interaction domain that people know about interacting with proteins. So why is this so important? Well, it's because you can now use this for the so-called 2-hybrid screen, or often people call this the yeast 2-hybrid screen, because you use Saccharomyces cerevisiae for this. Critical to have this work, these two domains have to function independently of each other. So we're just taking two domains. Again, usually there are more of them, but the most simplification is you've got a DNA binding domain and an activation domain. Those can function separately, so you can actually separate these two domains from each other, and you've got a DNA binding domain and an activation domain. And as long as these two things are together, you actually will get transcriptional activation, but you can also separate them from each other. And so what you can do is you can take one activation domain, I should say, from one protein, put it onto a DNA binding domain from another protein, and vice versa. And we talked about this a little bit before. Basically, you can even have a E. coli DNA binding domain, and you can put a yeast activation domain. So eukaryotic and bacterial together, they still work actually perfectly well. So now what you can do is you can take your activation domain and move it to a different part of your DNA just by attaching it to a DNA binding domain that will bind wherever you particularly want it to be. So just as a model of this, here you can have a DNA binding domain. Here's your activation domain. If they're not attached to each other, then you don't have transcriptional activation. So that's what you see. However, if you can somehow stick these two things together, which you can do through two different proteins, now these could be completely different domains. We've got our semicircle domain here and our globular domain or spherical domain here. These two come together, now you get transcriptional activation. So it's literally the yeast 2 hybrid screen is a way of looking at protein-protein interactions. What you're looking at here is the interaction between this semicircle and this circle. Of course, they're in 3 dimensions as well. The way this is described in the textbook, which I think is a little too complicated, so I did a little bit more of this background, you have a DNA binding domain over here and a DNA activation domain over here. So the blue one and the green one. Now what you're looking for here is an interaction between these two different domains. And usually the way this is done is you have one particular protein that you are interested in what it interacts with. So you've got some genetic data that says, this protein is critically important for, let's say, mating type switching or the cell cycle or something like that. And you want to know who or whom it's interacting with. So you take this is what's called the bait domain, this you know, and then you go through lots and lots and lots of different fish domains. And so this should be multiple different colors here. This fishing domain over here usually will be your complementary DNA from some kind of library. So you know you've got 2 interacting proteins present in one particular cell type. You get all of the messenger RNA translated into cDNA. Use that as your fishing domain. One of the reasons that people do this in yeast is there are lots of great genetic tools in yeast that you can use to make sure that this actually works. So the idea here, eventually what you want to get, you've got an upstream activating sequence that's going to bind to your DNA binding domain, a reporter gene over here that's only going to be activated when you have interactions between DNA binding domain and activation domain. This will assemble the whole pre-initiation complex and do its thing. How do you have that happen? you have two separate plasmids that you have to put into this yeast cell. One of them has the bait gene, again, this is the one that you know and you want to see what's interacting with it, connected to a DNA binding domain with a selectable marker. In this case, is a tryptophan marker. You have a separate plasmid which has your activation domain and your whole library. So this is going to be a whole set of different vectors. This is a different selectable marker on it. You take these two, you put them into a yeast cell. First, you have to have one that needs both of these selectable markers. So it's a yeast cell that doesn't have the tryp gene, it doesn't have the lu gene, and then your selectable marker down here, which is your reporter gene, which in this case is the histidine utilization. It doesn't have to be any of these genes. People use lots of different ones. But the nice thing with all of these auxotrophic markers, i.e. the cell can't grow unless you give them these things, is that it's a nice binary output. Grows or doesn't grow. If it grows, then you can go back in and isolate the plasmids, particularly this fish plasmid, and say, oh, the cDNA that I found in here interacts with, or the product of it, I should say, interacts with the product of this bait gene. And so you can do this. You can do hundreds and actually literally thousands of yeast colonies that you've transformed with your bait gene, and then individually with different constructs from your library, and then you pull out the ones that are making colonies. To do this properly, you also then have to switch this around. Now you found a gene over here that was your fish protein. Now you exchange it, put your fish protein over here, and make sure you pull out the same bait again to make sure that you've really got specific interactions between these two different things. And so this is, again, a great way of showing that at least inside a yeast cell, you've got two different proteins that are interacting with each other. And it's a wonderful assay. You can screen through, again, literally thousands of genes. And they don't have to be yeast genes. It turns out that activators and repressors, those are the ones that are working in yeast, but the protein-protein interaction can be human proteins, they can be plant proteins, you name it. They all seem to work. So questions on yeast 2 hybrid before we move on and talk about another whole group of DNA binding proteins, transcriptional regulators. Okay, so let's talk a little bit about the nuclear-receptor proteins. These used to be called nuclear hormone receptors because most of them bind to hormones. But multiple different things, multiple different translational regulators, and very important for development. This is, in fact, the first reason that people started to study these things. And it turns out that they have 3 conserved domains. One, a DNA binding domain. One, a ligand binding domain. And in most cases, again, this is a hormone. And then a very variable part of the protein, usually at the N-terminus of it. And the fact that you've got these 2 domains, DNA binding and ligand binding, that are very similar between all of these different proteins probably means, again, that they arose from one original progenitor. What do these guys bind to? Usually, these are relatively small molecules known as hormones, and then, hormones, so if you've had general biology and maybe even A&P. How many have had A&P or are taking A&P now? Okay, you know more about hormones than I do. But the basic idea here is that these are signals that can spread throughout the whole body, your whole system, and then have different effects in different places. And so the way that those effects actually take place is you have hormone binding by one of these hormones to one of these nuclear receptors. These then will bind to DNA, and depending on that nuclear hormone receptor, they can either be repressing or activating. These guys bind to specific so-called response elements. I hate the term response elements, but that's what's been used, because if you take this particular DNA sequence and put it in front of a reporter gene, then you'll get a response to a particular hormone. And so that's where that nomenclature comes from. These are various different response elements for different hormones. Here's the glucocorticoid hormone response element, estrogen response element, et cetera, et cetera, et cetera. These have very interesting structures if you just look at the DNA sequences of these. Two different kinds, and we've already talked about the two different kinds of DNA structures you can have. You can either have inverted repeated sequences or direct repeat sequences. And these are really typical for these nuclear hormone or nuclear receptor protein binding sequences. So here's a classic one with an inverted repeat sequence on the 5' AGA-ACA, 5' on the opposite strand, AGA-ACA. Also separated, as you'll see here, by almost exactly 10 nucleotides, which means that these are actually on the same side of DNA. So that nuclear hormone receptor is going to be binding on one side of the DNA. And it binds to 2 monomers of this protein, which are now head to tail. And just also, so the DNA binding sequence is an inverted repeat. The dimers are binding in a head to tail fashion. This is what you find in, again, the glucocorticoid response element, estrogen response element. There are a number of other nuclear hormone receptor proteins that bind to direct repeat sequences. So again, this is now on the same side, AGA-TCA, AGG-TCA. And what happens in those cases, instead of your protein binding head to tail, it's going to, head to head or tail to tail. Sorry, I should, have to go back and rewind that. So head to head or tail to tail is what happens in your inverted repeat sequences. It's the head to tail that happens on these direct repeat sequences. So if you just look at where my arms are relative to each other, here where my wrists are, it's going to be binding in the same place with the same sequence in the same direction. If I turn it around, it's going to be binding the opposite direction. So left and right. So just that's that symmetry that we're looking at here. So again, these guys inverted repeat. These are going to be head to head or tail to tail. And then the direct repeats are head to tail in terms of how they're actually binding. And you can see this here. Almost all of these binding sites are palindromic, i.e. read the same forward and backwards. So AGA-ACA and AGA-ACA. And that's just another way of talking about these inverted repeat sequences. Estrogen response element in the same way. So how do we know about what these things are actually doing? Lots of really nice cell biology, but it's also taken advantage of some of the reporter genes that we've talked about before. Why are they called nuclear receptors? Well, it's because they come and go in and out of the nucleus. Yeah? Yeah. So the question is, if you're going to have a palindromic sequence, can you have random nucleotides in between? Officially, for a palindrome, they should actually be opposite on opposite strands. So that would be a complete palindrome. Here it's just the actual binding sites, which are palindromic relative to each other. Okay, so nuclear hormone receptors. Why do we call them nuclear hormone receptors? Because, again, you find them in the nucleus. And for many of them, particularly things like the glucocorticoid hormone receptor, what happens when it binds to its ligand is then that complex gets transported into the nucleus. And it's that transport into the nucleus which leads to your transcriptional regulation. And the way that that was shown was by, again, using reporter genes. And here we're using a ligand, not critical what it is. It's basically a ligand which is really similar to the glucocorticoid hormone. If you have a fusion protein between beta-galactosidase and the glucocorticoid receptor, in the absence of hormone, this protein is present in the cytoplasm. In the presence of hormone, it's in the nucleus. It turns out you can do this just with that ligand binding domain, which is what we talked about. We talked about the multiple domains that you have of these proteins. So you don't need the whole protein. all you need is this ligand binding domain to direct whatever protein it's attached to into the nucleus in the presence of hormone. So what people think is happening here, this is your classic nuclear receptor, an activation domain, and a DNA binding domain, and a ligand binding domain. This would be a wonderful transcriptional activator and would turn on all kinds of different genes if it were in the nucleus. But it's not. And it's held out in the cytoplasm by a number of inhibitors. The book gets into excruciating detail about the names of all these things. I'm not using them in lecture because I'm not going to be asking you all those names. I get confused after about 2 minutes. So you guys can probably hang on to it for at least 5, but that's still probably not enough. So these inhibitors, take cell biology, you'll learn a lot more about the different ones. These inhibitors are present again, keep that protein in the cytoplasm until you have the hormone binding by the ligand binding domain. Hormone binding in ligand binding domain causes a massive structural change in that particular domain, no longer associates with the other proteins, and you now have the glucocorticoid receptor, which translocates in the nucleus. Once it's in there, it binds nicely to DNA and will activate the genes that are right next to it. In some cases, turns out in a few cases, depending on where that binding site is, it actually overlaps with the promoter. It can block any kind of assembly of your pre-initiation complex, et cetera. Because if you remember, hopefully from previously, it's a really big complex if you think about what's going on in the pre-initiation complex. It's got polymerase, it's got mediator, it's got all the general transcription factors, et cetera. So you have to have a really big complex associating with the DNA here. You can also see nicely in this model structure that this is a inverted structure because we've got our activation domain over on this side, activation domain on this side. So just by looking at this particular structure, you can tell there's an inverted repeat sequence that it's binding to because that head-to-head interaction. Okay, any questions on glucocorticoid receptors or nuclear hormone receptors in general? Again, this is the kind of level I expect your understanding to be, not the level of detail of all those individual proteins that they go through in the text. Yeah?
So is the response element actually on the DNA that's being transcribed then?
Yeah, so the question is the response element on the DNA which is being transcribed? The answer is usually yes. Now, it turns out that these are sometimes enhancers, and so they can be really long distance away and sometimes on other molecules. But for the most part, particularly in something well studied like yeast, those will be very close to the promoter which they're regulating. But again, that's most cases. We know of some cases again with these enhancers where they're 250 or even as much as 500,000 base pairs away. But usually they're pretty close. Yeah? Sorry, I'm not sure I'm quite following here. Oh, the proteins underneath them here? Okay. Yeah, so I guess the question here is, does each of these cartoons here correspond to what's going on up above them? Yes, they're supposed to be corresponding that way. Exactly. Okay, switch gears completely and now talk about the DNA. So hopefully Eric Iverson talked to you a little bit about DNA methylation and how that's important for interaction with the polycomb proteins and shutting down your transcription. But the way that's happening is happening because of the compaction of that DNA into heterochromatin so that your big pre-initiation complex that I was just talking about can't actually access the promoter. So there are some other kinds of things that happen with DNA methylation, and probably one of the most bizarre, because we don't quite understand why it does it, but it actually does happen, is something called genetic imprinting. And genetic imprinting is a DNA methylation that gets passed along from generation to generation. And DNA methylation, as again we've talked about probably almost ad nauseum, or at least Eric hopefully did when he was giving his lecture, is usually causing, again, this shutting down of the DNA. And so if you've got DNA methylation, particularly at promoter regions, you're now going to have compacted chromatin, so it's not going to be working anymore. However, we know of a number of cases of DNA imprinting. Again, this is methylated DNA that gets passed along from generation to generation, where it actually stimulates the expression of some genes. And at first, this is like, wait a minute, DNA methylation is supposed to be blocking activation of genes. Well, it just depends on what you're actually going to be methylating. And so in this particular case, this is for the IGF-2 gene, you have an enhancer which acts on this gene, but it can only activate if you don't have this insulator element between the two. And so insulator elements, did he talk about insulator elements? Hopefully he did. But the idea with an insulator element, this is sort of the opposite of an enhancer. So you've got something in between genes which will block the activity of the enhancer. It's sort of also usually these are scaffolding proteins, and so they're going to be binding that particular piece of DNA to part of the nucleus. And so an enhancer on one piece of DNA can't act on even something on that same piece of DNA because it's got to get past this scaffolding protein. So these insulator elements are bound by these scaffolding proteins. However, if you have methylated that particular insulator element, you no longer can have binding by this scaffolding protein, which then allows the enhancer to act. And so here we have a case of DNA methylation down here at the bottom. Turns out it's paternal that you have this, and it's always in that paternally inherited chromosome. In this case, you've now got activation due to DNA methylation, but it's DNA methylation clearly at a different place than you have binding of the enhancer and the IGF-2 genes. It happens, so again, I wanted to talk about it. We don't know why it exists. It's just in placental vertebrates, or as far as we can tell, mostly in placental vertebrates. Probably has to do with some kind of cool virus infection that happened way back when. No idea whether that's true or not. You get imprinting on either male side or female side. Again, it's just how that methylation takes place. Maybe about 100 genes, but still some of them which are really critical for development. So if you have problems with these insulator elements or some of the genes that are going to bind to that, and in fact for IGF-2 this is really quite well shown. IGF, by the way, stands for insulin-like growth factor, which gives you an idea what it's important for. Again, development and growth. And so if you have mutations in some of these genes, they can cause some real problems in terms of development. So the last thing I wanted to talk about, at least in terms of the methylation and epigenetic inheritance, is probably the best known epigenetic inheritance, which is X chromosome inactivation. So X chromosome inactivation is something that has to happen in, well, say, X chromosome modification has to happen in those parts of the population which are genetically superior because they've got 2 copies of an X chromosome. So males only have 1, so we're deficient. Usually just having 1 of these is a problem. Again, all of the autosomes, you've got 2 copies. And you actually do need 2 copies of most of the chromosomes and most of the genes that you have. So if you're missing a chromosome, and in fact this is one of the reasons that people do prenatal diagnosis, you can see that there's missing 1. 3 chromosomes is okay. Trisomy 21, for instance, Down syndrome. These are organisms that survive. But it's very rare that you can have a haploid and still survive. So that's why meiosis has to work terribly well. So you have to have some kind of mechanism to deal with X chromosomes being out of balance between males and females. And particularly important for males or females. And there are, of course, multiple ways to deal with this. And these are the mechanisms called dosage compensation. In fruit flies, all that happens is the X chromosome is super active. Get twice as much production of all the genes on the X chromosome in flies than you do in females. So, yeah, that's one way to deal with it. Another one is to downregulate both copies of the X chromosome in females. This is what happens in C. elegans. And in humans, and again, most placental mammals, one of the X chromosomes in females is completely repressed or almost completely repressed, whereas the other one is normal. And so that means that in females, particularly female humans, but again, all mammals, you've got expression from one of the chromosomes and no expression from the other X chromosome. And as we all know, there's the beer drinking, TV watching, and radiator gene on the Y chromosome, which are generally useless. So, so how does this work? Again, already mentioned, males and females have different numbers of chromosomes. There are a lot of genes on the X chromosome. Actually, one of the largest chromosomes in mammals is the X chromosome. So you do have to have a pretty sophisticated mechanism for dealing with, again, case of what's happened in mammals, completely inactivating one of those chromosomes. Turns out that inactivation, not surprisingly, happens by chromosome condensation, and you can actually see these in cells just by looking for the X chromosome, and you see they're really heavily compacted, at least one copy of them, in female cells, and in male cells you never see these particular bar bodies. How does it happen? Well, just in cartoon form here, early embryo actually has both. And so an embryo which is going to develop into a female mammal has two active copies of the X chromosome. And in fact, having those two active copies is really important for all that sex determination that happens. And so if you've got two active copies early on in development, that will lead to a number of cells then becoming the female specific cell lines which happen. But after about 1,000 cells, so you can do the back of the envelope math, figure out how many cell divisions that is, then you have condensation of one of the two X chromosomes that this female's inherited. It could either be the parental chromosome or the maternal X chromosome. Once this happens, however, this is epigenetically inherited. So if you've inactivated the parental X chromosome in this particular, again, one of those about 1,000 cells, everything that's come up derived from that particular cell will be expressing things from the maternal chromosome, and again, on the flip side, paternal chromosome here. The classic example of this are calico cats, which have multiple different colorations dependent on which of these particular chromosomes got inactivated. Also, one of the reasons why you very, very rarely see male calico cats. They're almost always females, because if you had a male, then you're going to have just one X chromosome, and you have lots of problems with that haploid case there. So if you look at mice, it's just a different way of looking at this. Again, it's a coat color gene. The reason people like to use coat color genes is because A, they're on the X chromosome, and B, they're really easy to notice in the adult animal. So here, both X chromosomes present and active in a precursor cell. One gets inactivated in each cell. So if you have a mutant coat color here and a wild type coat color here, you'll end up with a mosaic of the two different kinds of cells. And this is, in fact, what's used usually when you're going to be making ES cell knockout mice, is you'll take advantage of this, and in the females, find the ones which actually have this mosaic phenotype, and that'll tell you that you've gotten your breeding that's taken place appropriately. So it's a very useful tool as well. So we've known about this actually for a really long time, probably at least as long as people knew about bar bodies and microscopes, so literally a couple of hundred years. But it's only been the last 10 to 20 that we figured out exactly how this happens. And there's still a lot of questions about exactly what's happening. So what seems to happen is that you have expression of a couple of different RNAs in the X chromosome. And so it's an RNA which is encoded on the X chromosome. And there are really two different ones of those. There's the XIST and the we'll look at the other one just a second. There's also the T6, and these really act in opposite ways to each other. So you get synthesis of this particular RNA, this XIST RNA in the chromosome, which is going to be inactivated, binds to a number of different proteins, including those polycomb group proteins, which then cause DNA methylation. And that DNA methylation also gives you hypoacetylation of your histones and gives this whole compaction process. You also have exchange of histones. There's an alternative histone which is also used in these inactivated X chromosomes. And so you start out again expressing XIST RNA, and that gradually coats the whole X chromosome and causes this massive compaction to take place. I don't like this. It should really be much more compacted here as well. So if you look at zoom in to this X-inactivation center, here's a mouse X chromosome, but humans are basically identical to this. The XIST RNA is made, curiously enough, from a number of different exons, so it actually gets spliced as well. And then on the opposite strand is the T6 promoter. And the T6 promoter which is happening. And basically, it seems to be a balance between the two. If you have more of T6, then you're not going to be having inactivation of that chromosome. More of XIST, that's going to be the chromosome which ends up getting inactivated. So let's look at that again here. Here we've got the two X inactivation centers, by the way. That's what XIC stands for. They pair with each other. XIST is transcribed on one, T6 on the other. Why this actually happens and how it happens, it may just be completely stochastic or random. There doesn't seem to be any rhyme or reason to why one or the other of these X chromosomes happen. So XIST RNA coats, and this is actually probably together with those polycomb proteins as well, coats the chromosome, leading to inactivation. It's interesting that this expression only coats that one particular chromosome. Why it does one and not the other, it's probably just they're slightly closer. It's like wherever that XIST transcript is made, it'll complex with the proteins, complex with the chromosome, and then stimulate the binding of XIST right next to it, even though these guys are paired with each other. After, again, you get this binding, then you've got the polycomb proteins, which lead to the complete inactivation. When you actually have XIST, which is being expressed, it turns out that it's not terribly compacted, and so you have to have all of these polycomb proteins, et cetera, which will associate with that chromosome in order to get inactivation. And you can kind of see that here. Here are some fluorescence microscopy images. If you just look at XIST RNA, now this is in an already compacted chromosome. Now you look at the X chromosome itself. This cell now has two X chromosomes. XIST is associated with one and only one of them. And that interaction is, as far as we can tell, also dependent on this really small transcript, which is inside the XIST transcript. Both of those together seem to lead to that localization, which is here. And again, it's that localization which happens. And here you can tell that the X chromosomes are not terribly compacted yet. No, it's only now after the interactions of all these other proteins which is going to lead to the full compaction that takes place here. Here's an X chromosome which is not compacted, not associated with XIST. Here's the one which is in the process of being compacted and associated with XIST. It seems, again, to be random, as far as we can tell. No way to tell which one is actually going to be inactivated. Turns out it's actually not the entire chromosome. Now, why would that have to be? Any ideas? Yeah. So, still have to potentially replicate, which is a good answer. What else do you need to do? What do you need? What's critical for getting it? So remember, this is inherited, so from cell to cell. So after you do replication, what has to happen? Pardon? Well, you have crossing over that takes place, but what's causing that inactivation? What's the first step? Expression of the XIST RNA. So you need to be making a lot of this XIST RNA. To do that, you can't be completely compacted. And so the region right around the X-inactivation center is not compacted. It's actually cranking out this RNA. Because every time when you undergo cell division, you need a lot of that RNA to coat that particular chromosome and lead to the complete inactivation. Yeah?
If there's a functional mutation in one X chromosome, will the other one be activated and deactivated to switch over?
So I think the question here has to do with mutations that happen, which would either inactivate an inactivated chromosome and activate an inactivated chromosome. I think that's sort of paraphrasing your question. Not really.
So no, so if there's a mutation on one that's there,
yeah,
will the body actually, if the mutation's on the active one, will the body inactivate it and then?
Oh, so the question is if there seems to be some kind of deleterious mutation on one of the chromosomes, is that preferentially inactivated? As far as we can tell, no. Probably just what happens is in those cells where that chromosome is not inactivated, those cells are no longer functional. Okay, and then of course, it's only one way to skin the cat or make the calico cat. There's lots of other dosage compensation that happens, and if you're interested, send me an email. Dosage compensation in plants is totally cool. But again, I'm not going to get into it as far as this class is concerned. Okay, with that, perfect place to end. Well, not quite end, but getting close there. What happens down at the 3' end of your RNAs? As we know, for eukaryotic cells, there's a polyA tail. But how do you get a polyA tail? Well, there are a couple things that happen while you're making a messenger RNA. And so that's supposed to be shown up here with this blue and green line. This, of course, is part of a much longer messenger RNA. In eukaryotic messenger RNAs, there's a consensus sequence here, AAUAA. The sequence is not critical. But what's important here is that this is a binding site for a particular protein, which is called the cleavage and polyadenylation specificity factor, i.e., so it binds to the RNA. Again, so this is the RNA sequence. This should sound kind of familiar as far as row-dependent termination in bacteria. There's the RUT sequence that row protein binds to. Here we have the CPSF binding site. And then 10 to 30 nucleotides later, there's a cleavage of your messenger RNA that takes place. And this is now the template for polyadenylation by the polyA polymerase. To get this to take place, this cleavage to take place, you also have to have binding by this cleavage stimulation factor, which is about 30 nucleotides downstream of this binding and cleavage site. And this is now different from the case that you have with row-dependent termination, where basically that row protein pulls the RNA out of the RNA polymerase. Here, you just have cleavage of this RNA and then the polyA tail. Well, what happens to this green piece right here? Well, it turns out that there are exonucleases which chew in at the end of this. And hopefully you talked about cap sequences, what happens at the 5' end, which protects that end. Well, there's no cap on this particular piece of RNA, so it gets chewed up. And it turns out that that exonuclease activity is probably what leads to termination. So you just chop up all of this DNA, the RNA polymerase is here. That then basically chops up all the RNA which is there, nothing for the RNA polymerase to bind onto, falls apart, and you can start again. Just shown in a slightly different way here. Again, binding in your RNA as it's being transcribed. It's not right here at a termination site. It's just some sequences that are in the RNA sequence which are there. You have binding by CPSF and CSTF. But wait a minute, they're not bound to the RNA here. What are they bound to here? What's this funky light blue thing hanging off the RNA polymerase? It's the C-terminal domain, highly phosphorylated C-terminal domain. So these guys are coming along for the ride. They're basically waiting until the polymerase transcribes the appropriate RNA. And so that means it can be very efficient. These proteins are right next to where that RNA is being made. So as soon as one of these sequences is made, you can get this binding, and then cleavage will take place. That RNA is chopped. The polyA polymerase comes down, binds to this, makes a whole stretch of A residues. Those get bound to by the incredibly creatively named polyA binding protein, and that will give you your final mature RNA. Usually you end up with about 200 or so of these A residues at the end of your RNA. It varies from species to species, but in humans it's about 200 to 250. And these polyA binding proteins will bind to between 5 and 10 nucleotides a time. And it's not just the polyA tail. Everybody says, what protects the 3' end of eukaryotic messenger RNAs? PolyA tail. Well, it's not really the polyA tail. It's the polyA binding protein, which is protecting the 3' ends of your messenger RNA. So there are a number of other proteins which are involved here, but the main ones, again, that we talked about are the cleavage polyadenylation specificity factor. This is the guy that's hanging out on the C-terminal domain. Transfers as soon as you make one of these specific sequences. This cleavage stimulation factor binds further downstream of that, leads to the actual cleavage itself, which we're not talking about, so you can ignore these two. PolyA polymerase, this is what adds those polyAs. So this is now a non-template dependent RNA polymerase. So it just takes ATP and adds a whole bunch of A's onto the ends of your messenger RNAs. Once it's made it, then we have polyA binding. And it turns out that polyA binding, the polyA binding protein binding to that polyA tail, also stimulates the activity of the polyA polymerase. So polyA binding protein binding to polyA will lead to more of this polyA polymerase activity. And so if you have the lack of your polyA binding protein, then you don't actually make a really long polyA tail. This seems to be sort of a quality control process. So once you start with that polyA tail formation, as long as there's enough polyA binding protein around, then you can continue to make the rest of it. So this is another way of looking at this. Here we have our binding site for CPSF, which is a complex of multiple different proteins. Binding site for your CSTF, and then association of the polyA polymerase. PolyA polymerase extends a little bit. You have polyA binding protein, extends a little bit more polyA binding protein, etc. And you end up here with, again, about 200 nucleotides that you have at the end of your messenger RNA. So now we're done, right? Well, actually, no. We need to get this RNA, once you've modified that 3' end, out of the nucleus, because almost all the translation, or all the translation, people argue about this, takes place in the cytoplasm. It doesn't take place in the nucleus. So you have to now get this RNA out of the nucleus before getting it translated. So let's talk a little bit more about that. Now, if you think about the structure of the nucleus is a membrane bilayer. It actually kind of looks like another cell inside the eukaryotic cell, which has a bunch of pores. So this is down here is your nuclear pore complex in this bilayer. This nuclear pore complex is, in fact, one of the largest macromolecular complexes that you find in a cell at all. Seems to be on the order of outside of a couple of ribosomes altogether, and it's important for moving things in and out of the nucleus. Probably the most important is going to be your messenger RNAs. Now, it turns out that the nuclear pore complex is way too small to be able to put a whole messenger RNA through in a diffusion mechanism. And so what you have to have are these messenger RNAs which are being made, and so that's just cartoon-like here. So this is your DNA template, which is the long string here, and each of these blobs coming off of it is your messenger RNA. Well, why is it a blob rather than a nice little messenger RNA? Well, that's because of all of the modifying proteins that are associated with this RNA as it's being made. So you have all your capping complex proteins, you've got your splicing proteins and RNAs, and you've got what we just talked about, all of the tailing proteins, which are associated with that RNA, including the polyA tail. And so these are the so-called heterogeneous nuclear RNPs, so heterogeneous nuclear ribonucleoprotein complex, and that's what needs to be transported out through this nuclear pore complex. So this is this big blob here that actually seems to have to be stretched out and actively transported through the nuclear pore complex. Once it's been transported, now the ribosomes can associate with that messenger RNA and all of those translational initiation factors, which we all remember, right? Yes? No? And leads to translation taking place of that messenger RNA. And one of the things you'll notice here in the cartoon is that we've gone from this big blobby structure here to basically a pretty naked RNA structure here. And it turns out that this is really important for getting your translation. You think about the ribosome, it's clamped down on top of the RNA, right? And so you have to have removed a lot of these other proteins in the hnRNP before it actually comes out. You can see this under an electron microscope. Here's these hnRNPs. Here's a nuclear pore complex. And here's that hnRNP being put through here. All of these black blobs here actually represent ribosomes. And so they're now translating this RNA as it comes through the nuclear pore complex. So let's zoom in, as it were, at least look at where a lot of these proteins are. And so this hnRNP has that poor little RNA in the middle here, little red thing that you can barely see. Well, that's because, again, it's coated with all of these different proteins. So first thing, and probably most critical, you're never going to get an export of an RNA if you don't have appropriate cap structure. Because if you don't have an appropriate cap structure, all those initiation factors, translational initiation factors I talk about, can't associate with anything. They don't know where to start. Because remember, it's that cap and the cap binding complex that you have in translational initiation. So you have to have the cap binding complex here at the 5' end. You also need your polyA tail with all of this polyA binding proteins down here at the end, but there's all of this other stuff in the middle here. What's all this other stuff? Well, a lot of it is are the proteins which are involved in splicing and determining the appropriate splice junctions. So if you remember, one of the really neat things that can happen in eukaryotic, and particularly in the larger eukaryotes like us, is you have lots of alternative splicing. Now, if you're going to do alternative splicing, this is great, but you have to make sure that all the alternative splicing has taken place, and it's the right alternative splicing that's taken place. So a lot of the proteins that you see here, and again, they're just named, NXT, blah, blah, blah. It's not important what they are, but really important here are these exon junction complexes, and that's what these are particularly named here. Exon junction complexes says, hey, we've put two exons together. They're right. We have now this intron removal that's taken place. Because if you export a RNA that's got a bunch of introns in it, whatever you translate is going to be a god-awful mess. So making sure that you have all of these appropriate exons joining together are really important. You also have these SR proteins. Did you talk about SR proteins, hopefully, in lecture? They're really important for alternative splicing. Which of the splice sites gets used? SR proteins then either use or don't use one particular intron or exon. And so these guys are all associated with your messenger RNA. It then gets transported through, and in fact, in that transport process, you lose a whole bunch of these proteins. And a lot of these proteins are then recycled back into the nucleus for the next messenger RNA that you're going to need to export, get out into the cytoplasm. And then it's this complex with our friend eukaryotic initiation factor 4E binding to the cap structure, which now can be translated into your protein. Other thing which, again, hopefully you remember is that these polyA binding proteins also associate with the cap structure, and that's really important for stabilizing your messenger RNA while it's being translated, but also allowing the ribosomes, once they get to the 3' end, to associate with the 5' end and go back and cycle through this whole process. So now you've exported your messenger RNA. Now that messenger RNA can get translated, and we make all of our proteins, and the cycle begins again. Questions about RNA processing and export? Otherwise, I will review the guest lectures on Friday. There will be clicker questions based on today's lecture, and enjoy the sunshine. Yeah. Oh, yeah. Real quick question. Sorry.
Those polyA binding proteins
yes.
actually stay attached to the mRNA when it comes through?
So the question is, do the polyA binding proteins stay associated? The answer is yes and no. So polyA binding protein 1 stay associated. PolyA binding protein 2 doesn't. But as far as we're concerned for this class, yes, they do. Pardon? The pores are size-specific, you mean? Sorry.
Yes.
The
pores in the nuclear membrane,
yeah.
And I'm assuming that the external membrane as well, the cell membrane, they're size-specific?
Yeah, well, there's both. There's size-specific and also, and I didn't talk about any of the export proteins. There's a whole bunch of other things which give you specificity in terms of that process. And so that specificity aspect has to do with, and so it turns out the nuclear pore is actually too small to get one of these messenger RNAs complexes through, so it has to be associated with other proteins, and basically those other proteins get transported, and basically they pull through that whole complex. But I didn't talk about that today. I think they'll talk about more in cell biology next term.
Okay, because I'm thinking about electroporation,
Mm-hmm.
and that specifically is, well, it's the external membrane, but that's the size-specific.
Yeah, so electroporation, but that's a completely physical thing which is going on here rather than a biological process.
But you wouldn't be able to have a lot of organelles crossing over the membrane because they're too big.
Yeah, most stuff is too big. And in fact, the messenger RNA complex is too big, and the only way it can get through is by very specific transport.
It does actually go through, in the picture it didn't look like it, did it?
It just looks like it gets smushed and then it squeezes the RNA out.
Yeah, what actually it does, and so that was what that final process was supposed to be showing, was that you actually, in the cartoon anyway, there's still a lot of proteins that are associated with it. And so kind of this guy's question about the polyA-binding protein, yeah, the polyA-binding protein is still associated with that messenger RNA when it comes through. It's just that it's not quite as humongous, but there's still quite a few proteins that are associated with it.
I have 2 questions I wrote
Yeah,
down.
sure.
So one of them was, you kind of asked the question, but it wasn't the correct question. So
Yeah.
about the X-inactivation gene, if it's mutated, is that when you get 3?
Oh, okay. So what happens if you have problems with X-inactivation? Usually it's lethal. So usually if there are problems with X-inactivation, you'll never develop, that embryo will never develop properly. And so it's just something which spontaneously aborts or whatever is happening there. Just the development doesn't happen properly.
So you don't end up with, that's not how you end up with 3 X chromosomes.
No, and those are very, very rare. But usually that has to do with actually way downstream when you end up with meiosis at the very end and the formation of the egg cells. And so what will happen is then in some cases they'll actually be diploid as opposed to haploid. So partial diploidy, and so you end up with 2 X chromosomes that are in an egg, and that then gets fertilized by a sperm, and then you end up with 2 maternal and 1 paternal X chromosome there, and so that's how that usually happens, and that gets really confusing. But it turns out that inactivation can work in some of those cases. So you can actually get 2 of your 3 X chromosomes inactivated, and then that actually seems to be relatively stable.
Like when you have a child and they resemble one parent versus the other one, does that depend on if the maternal or the paternal X chromosome is inactivated? So like, oh, this kid looks like their dad, they must have more, That's stuff which is way, way, way beyond my understanding. So I have absolutely no clue, and people are looking into that. No, it does kind of make sense, but it only is going to be able to work for the genes that are on the X chromosome, some of which are important for development, all kinds of other different things. And a lot of these things are polygenic, and so it's going to be some autosomes, some, and so there are lots of autosomes. I have 2 girls, and it's hard to imagine how they could be more different than each other, even though it's the same, in theory, same genetic content which went into making the 2 of them. I think I was thinking about the calico cat, and I was like, oh, I wonder if this works for people too.
Yeah. So for that particular case, it's coloration. And coloration is actually nice because it's a single gene trait, and so you can actually look at it. And so it's single gene, it's on the X, and so in those very specific cases, yes, you can say that that's what's going on. But those are really few and far between. Yeah, it doesn't seem to work with humans that way. And in fact, most of our coloration has to do with melanin, so it's a totally different story, and that's all autosomal.
Like hair color and all that stuff, that has nothing to do with that.
Again, I don't know enough about it to give you a good answer, to be perfectly honest. There's a human genetics, in fact. There is a human genetics course that we teach here, so that would be a better place to ask that question.
All right, thank you.
Oh, certainly.
Sorry.
No worries. Yeah.
I just have a question about the final, like with the midterm, like all the last term
Yeah.
midterm material. Should we like primarily study what was on the midterms, or is it?
No. I would primarily study what's in this last section because it's basically like a third midterm. That's the way I treat it.
It's not going to be
Not comprehensive, no. Not at all.
Oh my,
That is very
Yeah,
good
so
information.
the only, sorry, I thought I made that obvious before. I will try and mention that on Friday. If I don't, please one of you bug me. But yeah, the final is not comprehensive per se, but there'll be stuff like today, as like, initiation factor, yeah, right. So EIF4E, yeah, okay, well, it's important now, and we talked about it in previous midterms, but it's the stuff that I'm talking about now. And so again, I sort of assume that you know that stuff, but it's going to be really as it's applied to the stuff that we've talked about. So I really consider the finals like a third midterm, rather than, rather than something which is comprehensive.
All right, awesome.
Yeah.
Thank you.
Yeah. Good.
So I had asked you a question before you went away for a little
Yeah.
while about non-mammalian hemoglobin,
Yeah.
and you recommended researching it because you weren't sure.
Yeah.
And I looked into it and the genes that control like marsupial hemoglobin,
Yeah.
and it seems like there's actually a similar duplication that occurs.
Okay.
So they still have not really the same type of fetal hemoglobin, but they have sort of an alpha and sort of a beta. It just occurred after the marsupial.
Oh, marsupial placental split. Oh,
Yeah.
cool. That's very cool. Yeah. So presumably there was an ancestral, and somewhere I think I've seen an image where it shows sort of how the original duplication of hemoglobin, or the globin genes, of course, you know, way before hemoglobin, because you've got myoglobin as well, and all kinds of other stuff which is going on. And I think it was, you know, supposed to be with of a fish-like organism or something like that. And then after that, then